everyone, welcome back. So remember in our last video we were talking about metabolites, and so this is a figure from your textbook that shows how the metabolites can be utilized. E. coli can use glucose in multiple pathways. So glucose is broken down into different precursor molecules. These precursor molecules can be used to produce amino acids or lipids, carbohydrates, nucleic acids, or they can just be broken down and release energy in the form of ATP. So let's look at the three central pathways that we talk about. So the first pathway is called glycolysis. Glycolysis splits glucose into pyruvate, and in that process, it generates ATP. The pyruvate then is going to move on into other pathways so we can produce a lot of ATP. The pentose phosphate pathway is the pathway that produces NADPH. NADPH is used in anabolic pathways, whereas the glycolytic pathway is a catabolic pathway. Pentose phosphate pathway is an anabolic pathway. The glycolytic pathway leads to the Krebs cycle. So from glycolysis, the molecules generated, the pyruvate, are going to be modified and eventually will enter the Krebs cycle, where we will oxidize them completely, producing a lot of reduced molecules that can then produce a lot of ATP, or help in the production of a lot of ATP. So respiration in a nutshell is the transfer of electrons from glucose to an electron transport chain. In aerobic respiration, oxygen is the terminal electron acceptor. This is what you learn in general biology, in anatomy and physiology, and this is mainly what we talk about in microbiology. The majority of microbes that we are dealing with can utilize aerobic respiration. There are some that use anaerobic respiration. So in anaerobic respiration, they don't use oxygen as their terminal electron acceptor. They're going to use some other molecule. They might use sulfur as a terminal electron acceptor and produce hydrogen sulfide. That would be an example of something they might produce. They also use a modified citric acid cycle, so TCA, tricarboxylic acid cycle, also known as citric acid or Krebs. I typically call it Krebs or citric acid. But they're not going to use oxygen. These are anaerobic organisms. In this process, they produce ATP, but it is much... Whoa, that was weird. Less ATP than in aerobic respiration, okay? And then we have fermentation. Fermentation is an anaerobic process. It is basically the glycolytic pathway that is running, but it has to run continuously. So glycolysis is the oldest pathway that we have. And pretty much all organisms can utilize glycolysis. Some use glycolysis only as their mechanism of energy production. So fermentation is a mechanism of keeping that glycolytic pathway running. So at the end of glycolysis, we have a pyruvate. We have two pyruvate, and we have two NADH. So the NADH have to be oxidized back to NAD so that they can then help in the breakdown or the oxidation of another glucose molecule. And so we use a fermentation process where we take the hydrogens and we add them to pyruvate, and we produce different byproducts. And I'll talk about that a little bit later as well. Pyruvate, or I won't say that. Fermentation produces a net gain of approximately 2 ATP. These are all theoretical maximums. Aerobic respiration produces a net theoretical maximum of about 38 ATP. Anaerobic respiration produces less than 38 ATP, but more than 2 ATP. We don't know the exact amount. So we know that it's slightly better than fermentation, but it's not near as good as anaerobic respiration or aerobic respiration. So now let's look at how enzymes work. We talked about enzymes. They are biological catalysts. They are proteins in general. And enzymes are typically named for what they work on. Enzymes are very specific to certain molecules. They have an active site which typically binds only their substrate. So, which is how they become so specific. When the substrate binds, the enzyme changes shape slightly. This is known as induced fit. And the enzyme and the substrate respond to each other. The enzyme either builds or breaks bonds in the substrate and releases the end product for that reaction. Enzymes do not get used up in reactions, so they can work multiple times before they end up getting utilized, or before they end up dying or being broken down. Enzymes don't die. They're not living. They're organic material that isn't living. So here we have an enzyme, and here we have a substrate. The substrate fits in this region called the active site. When the substrate and enzyme come together, the enzyme gives it like a hug, kind of changes in shape, induced fit, which I talked about, either builds or breaks bonds within the substrate and then releases the product. And then this is probably a better depiction of what an enzyme and a substrate look like. Sometimes enzymes utilize cofactors or coenzymes. So a cofactor is an inorganic molecule, whereas a coenzyme is an organic molecule, but both assist the enzyme in whatever reaction has to occur. So I always think of cofactors, be it inorganic or organic coenzymes, as the helpers to the enzyme. So I used to tutor, and so this is my analogy of enzymes and substrates and coenzymes or cofactors. The enzyme is like your instructor, and they're trying to get you to understand the class you're in this case, microbiology, so that you can then leave the class with the knowledge you need and the grade you need to go on to the next class, whatever the next enzyme course is. The coenzyme or cofactor would be a tutor. Sometimes when an instructor speaks, the student doesn't understand what the instructor is saying. A lot of times people will say, oh, the teacher talks over my head. And in that case, you go to a tutor, and then the tutor can break it down more effectively. So I was the tutor at the time when I made this analogy, and I love this analogy because I made it, back in like 2004. And so I was that cofactor or coenzyme, the one who broke the information down and made it understandable to students. So the enzyme, the instructor would discuss something. I would make it more understandable for the students, and then the students, those substrates, were able to take the information and use it, and they were able to pass the class. And that's how enzymes work. So think about it. An enzyme is catalyzing a reaction, but sometimes the substrate and enzyme don't fit together. A cofactor, a coenzyme, helps them to work together. Examples of coenzymes: FAD, NAD, and NADP. These are all the electron carriers that I talked about in a couple slides ago. So what are some things that influence enzymatic reactions? Actually, this has been an almost 11-minute video. I'm going to stop here and we'll talk about this in our next video. Bye.